Cool. Hi, everyone. Welcome to BIOMES. BIOMES is YSE's flagship seminar presenting and bringing to you cross-cutting topics from environmental sciences. And I'm Alex Plusa, and I'm a fifth-year doctoral student, and I have the pleasure of introducing Dr. Javier Ceja Navarro for his talk, Exploring Microbial Trophic Interactions in Environmental Microbiomes. His research encompasses the complexity of host-associated and environmental microbiomes, including viruses, bacteria, archaea, fungi, and protists. And in the process, he unearths the natural history of microorganisms, how they respond to environmental change, or simply how they function. For example, he's been working extensively on the widespread agricultural pest, the coffee berry borer beetle, to identify the process of caffeine detoxification by gut microbiota. Dr. Ceja Navarro is currently an associate professor at Northern Arizona University. He previously was a research scientist at the Lawrence Berkeley National Lab after obtaining his PhD in biotechnology at the Instituto Politécnico Nacional in Mexico. And in addition to his scientific work, he engages in scientific communication and is committed to reducing barriers for underrepresented students. Dr. Ceja Navarro will present for about 45 minutes, and we'll leave time for questions at the end. Thank you, Javier, and for joining us today. And we'll get started.
Okay, can you guys hear me well? Okay, thank you. Well, thank you for being here. Thank you for inviting me, Alex, for nominating me, and it's a pleasure to be here to talk to you about some aspects of my research. As Alex mentioned, currently I'm in Northern Arizona University. I'm part of the Department of Biological Sciences, and I am also part of a research center within the university called the Center for Ecosystem Science and Society. And in my laboratory, we do what I just have there in the title. We explore microbial trophic interactions in any kind of environment, really. And what we do in my laboratory is that we try to combine traditional microbiology with multiomics. In traditional microbiology, what we try to do is to get an environmental sample and isolate organisms, all kinds of organisms, to analyze them, as you will see through the presentation. But then we also take environmental samples and then analyze everything that is in there by using next-generation sequencing. Are you guys familiar with next-generation sequencing? We'll get there in a moment. Okay, so I thought that I should start with describing what we do in my laboratory as multiomics. As you can see there, what I have here is a representation of an environmental sample where you will have bacteria, viruses, nematodes, protists, pretty much everything. You can go there and try to isolate those microbes, or you can extract DNA. You take the soil sample and extract the DNA of everything that is in there. From there, you can, for example, search for a specific gene among the DNA to analyze microbial diversity. Again, is sequencing. Or you can also chop that DNA into little pieces and then do sequencing of a metagenome. From the metagenome, you use computer work, bioinformatics, and then you try to put the pieces back to represent the initial DNA in your sample. Normally, it doesn't work perfectly. That's why I have the open rings in there. So that's what we do in the lab. Another strategy that you could do is to take environmental sample and analyze RNA to get an idea of who is active or working in your sample. Once you do that, you do a metatranscriptome. It's another form of sequencing technology. Finally, in the laboratory, we also extract proteins from samples, and with collaboration with national laboratories, we analyze the proteome, the proteins that are active in an environmental sample. And then from there, what we do is that we try to integrate all the data and create, for example, a network like what I'm showing here. Each color in that network shows the genome of a different organism and how they are connected in an environmental sample. So if you see, it's a massive stream of data. And it's not just doing it because, okay? we are always, or at least we try to go after a specific scientific questions, which is the idea of science in general. Now, before coming here, I had a little bit of a struggle, which is, what should I tell you about? Should I tell you about insects? Which insects? Should I tell you about my work with soil? Like which environmental process in soil? So for the presentation, I'm going to ask for your help now, in a way in which you are going to choose your own adventure right here. So first, I'm going to tell you about my work exploring the insect microbiome. And I have two options for you. We can learn, or I can tell you about a living bioreactor that degrades lignocellulose, or I can tell you about a coffee pest that is enabled by its microbiome. And now, people, I want you to vote, please. Which one do you want to hear about? The living bioreactor? Hands up, please. As always. The coffee pest? Very good. You know, poor passalid beetle. This is the same story every time that I attempt something like this. Everybody goes for the coffee pest. We all love coffee. So, but if anybody's interested about learning about the passalid beetle, we can talk about it after the presentation. Okay. So, okay, you all choose the coffee pest that is enabled by its microbiome. So let's go there. Allow me to introduce you to the coffee berry borer, or Hypotenemus hampi. This is the most devastating insect pest of coffee. And you can see a female beetle on top of a coffee bean. You can see that that organism is very small. The females will reach a maximum size of around 1.5 millimeters. And what they do well is that they are pretty much widespread in all coffee producing countries. And I bet you didn't know about it, right? Well, this coffee insect is pretty much destroying your coffee. Everywhere where coffee is being produced, you will find this beetle. And what females do is that they fly around the coffee plantations, find the coffee berry, create a little hole on one side, and then enter to the seed. Then in the seed, they start eating everything in there. They reproduce inside. They can lay like around 100 eggs, a single female. And then you will have, well, a reduction in coffee production that is around 80% when there is no treatment applied. Now, I will say a big problem about this insect and its control is that we don't have great strategies to get rid of the beetle. You know, nobody wants a pesticide in their coffee, right? You want your coffee organic and clean and nice. And we also care about our environment, and we try not to apply insecticides or anything, right? So what farmers do is that they go to the fields and they start picking the berries one by one. Anything that looks that is affected by the beetle will be picked and then burned. And that's a big loss, okay? But to me, that's not what makes this insect amazing, you know? I want you to think about the following. We have here an insect that pretty much spends its whole life cycle inside of the coffee seed. And there, it's just eating and eating, laying eggs, reproducing. But while doing so, it's exposing itself to toxic levels of caffeine. So imagine, this insect is eating around 10 hours through the day, and is eating enough caffeine that will kill any human when you put yourself in that size and in that ratio. Calculations are around the following. It is said that the beetle is consuming around the caffeine that is contained in 500 shots of espresso in 10 hours. So imagine yourself getting to drink something like that. Well, for that beetle, that means nothing, okay? But here is the thing. When you consume a lot of caffeine, I'm not talking about the 3, 4, 5 shots of espresso that we get every day, okay? Like toxic levels of caffeine, you can have all these problems that we have represented here. You know, you can have cell wall damage, damage of your DNA, impairment of protein traffic. And in general, I'm sure you know that caffeine is an alkaloid that is produced by plants to deter insects from eating the plant. So it's a defense mechanism. But for the beetle, this means nothing. So our question was, is the gut microbiome contributing to the process of caffeine detoxification? Are the microbes in the belly of the insect helping the beetle to be what it is? getting rid of this toxic caffeine. Back in the day, when I started this project, there were other scientists that were working in the project as well, and they were testing, for example, putting Drosophila into the same levels of caffeine as the beetle, and Drosophila will just die. They also took beetles that were phylogenetically related to the coffee pest. This is a bark beetle, a big problem, all bark beetles in general. And they tried to feed these beetles with caffeine, the same levels, and they just died, but not the CBB, which is how I'm going to call the beetle now, the coffee berry border. So again, the question that we have is around the microbiome. So the first thing that we did was to go and collect beetles from different coffee-producing countries, take a single beetle, extract its DNA, and then look for one gene, a gene to characterize the diversity of its microbes. This is what I want to show you here. Oh, I need to say this. This project was a collaboration between USDA and the Lawrence Berkeley National Laboratory. Now, this colorful image that I'm showing you here has a purpose, I promise you. What you have here are the beetles that we collected from these different coffee-producing countries. And then you can see them that they are connected to the different microbial groups that were identified as part of the microbiome of this beetle. What we found was that all the beetles shared some of these microorganisms that you see here. So they have a very complex microbiome. We did a statistical analysis and we found that the microbiome could change depending on location. You know, wherever the country was that you collected your beetles, they will have a slightly different microbiome. Still, they will share specific groups. But we also found that the microbiome of these beetles will change with coffee species. Do you guys know how many coffee species are there out there that we can consume as coffee drinkers? Well, two, thank you very much. There are the main two coffee species. The one that everybody loves, coffee Arabica, it has a content of around 20 milligrams per gram of caffeine. And then there is the other one, coffee Canephora or Robusta, that has a lot more caffeine, is a lot more bitter, and usually is mixed with your nice coffee without you knowing it. Nobody likes Canephora, but it's out there. Anyway, we could see that depending on what the beetle was eating, its microbiome will change as well. But well, you know, we wanted to go a little bit further in the analysis and try to do some molecular work to identify organisms that were important for the beetle, what we would call keystone taxa. So there I went back to my country. I went to Mexico, to the state of Chiapas, to collect hundreds and hundreds of insects from coffee Arabica and coffee Canephora, and then analyze the microbiome. And I did the same. You know, I took the beetles, amplified by one gene, identified the microbes with the beetle, and then did molecular ecological network analysis. Sounds complicated, it's not, but our results show the following. When we put together all the microbes that live in the guts of a beetle that is feeding on coffee Arabica, the communities may look connected in this way. But when you analyze the same, those microbes in beetles that are feeding on coffee Robusta, the community will look like this. So if you see, we have a little bit less complicated network here than when they are in Arabica. So maybe this is an effect of the amount of caffeine that is in the coffee, right? But the important thing that I want to show you here is that we identified specific microbes that are represented by these nodes as being important for the community, at least mathematically. Why are they important in the network? Imagine the following. Imagine that you have this microbe and you manage to get rid of it. What do you think is going to happen to the rest of the network? The network will disconnect completely. So that's the idea behind this network analysis. You identify those microorganisms that seem to be important in the network, and you can create a hypothesis and say, are they important for the beetle or not? So we identified keystone groups such as Pseudomonas, Stenotrophomonas, and Microbacterium. That's what I want to say now. So I was working this project started many years back. I had to stop for a little bit, but back in the day, you know, I was still working at the Berkeley laboratory, and I got a challenge by my supervisor in USDA, my supervisor in the Berkeley lab, and they asked me like, well, do you think that you could isolate any of those microbes and prove to us that they can degrade caffeine? Because maybe that will be the start of the project, right? So, well, how can I do that? Remember that I told you that the beetle is very tiny? 1.5 millimeters. So then the question was like, can you dissect a beetle like that and get the guts out, like very nicely dissected, and again, isolate the microbes? Well, so like challenge accepted. So I put together myself with a lot of coffee every morning, very fine tweezers. Coffee helps me. I'm usually very shaky, but so with very fine tweezers, and there you can see the process. You know, you have the tiny beetle, you open the elytra, which is the little shell that you see out here, then remove the membrane, the abdominal membrane, and then you can take the guts out. And here you can see the crop, pretty much you could say the throat of the beetle, the main belly, and then all the way to the anal cavity. I'll tell you, this was a big win, because back in the day, from among the scientists that were doing beetle analysis, nobody was able to dissect the beetle but me. Now I have a bunch of students that do it like this, you know, and it took nothing for them to learn how to do it. But okay, I was able to get the guts out, and then I prepared these kind of caffeine gradient plates. These are just agar plates that have caffeine as the only source of carbon and nitrogen. So there is not much carbon, well, agar, those that know, could be a source of carbon. But we started our plates with this amount of caffeine to this amount of caffeine. And I want you to see, guys, the trend that we see here. You can see that there are a lot of microbes growing in the area, but we have very little or little caffeine. And then as you move forward, less are growing, right? So back in the day, this was maybe 2014, I'll say, there were 2 caffeine-degrading microbes that were reported out there, 2 bacteria and 1 fungi. When we did analysis of everything that I isolated here, we isolated all these different organisms that you see there that maybe are just names for you, but each represent different species of organisms that we demonstrated that can grow in pure caffeine. Let me say this. After getting them in the plates, we take 1 microbe and put it in a pure caffeine solution. So we'll remove the agar, there will be nothing, just the caffeine. We put the microbe there, and they did well. They grew on pure caffeine. So then the question was, well, you have the microbes that can degrade caffeine. Are they degrading the caffeine within the body of the beetle? You know, there are two very different things, I'll say. So, there was a point here. I want you to see here these specific organisms that I'm showing you, the names of them. They are, according to identity of DNA, the organisms that we identified in the networks as important in the networks. So we were like, yes, well, we're getting something interesting out of here. So just look at these names. So then my supervisor told me, well, now you're great. You're able to dissect insects. You isolated the caffeine-degrading bacteria. Now show me that they are active within the beetle. Those are our good supervisors, okay? So what I did is that I collected beetles again from Mexico, extracted DNA and RNA. From the RNA, I synthesized cDNA. Just imagine, I'm using RNA because I'm looking for what is active. Let's remember that. And then I look for a gene. This is the first gene that acts on caffeine degradation. It's the first step of degradation in Pseudomonas. So what I did is that I did just PCR to see if I could identify it in the RNA. And I want to show you the results here. When you extract RNA, you are not supposed to get anything when you do PCR, okay? Because if you get any band in your PCR, your RNA is contaminated. So yay, it's clean, nothing amplified. But when you take that RNA and you turn it into cDNA, if your gene is there, you should be able to amplify something. And you can see there that the product is there. It was also in the DNA and in my positive. So that I took to tell my supervisor, guess what? I think that Pseudomonas is active within the beetle. Finally, you know, talking to my colleagues there in the laboratory, one said like, well, why don't you remove the microbes from the beetle? Yeah. Like, and then a chemist told me like, you could collect the frass of, do you guys know what's frass? The feces. That's frass, a nice way to say the word for insects. You can collect the frass from your beetles. Like, yeah, have you seen the size of my beetles? You could collect the frass of your beetles and compare the caffeine content in the frass and in the diet. And maybe you will see an effect of removing the microbes or not, right? So there are another problems, like how am I going to collect the frass of this beetle? right? A tiny, tiny organism. So I was attending a bunch of entomological society meetings there, and then I saw somebody that was using fluorescent spheres to track if a beetle was eating or not. Okay, I'll show you here. Let me go there for it. Yeah, thank you. Thank you. I hope you can see this. But what I did is that I prepared the food of my beetles. I added fluorescent spheres. And then here you can see the dissected guts of the beetle from the crop, the main stomach, that's not how you refer to it, and then all the way to the anal cavity. And if you see here in the arrows, I was able to see how the spheres that are in the diet that the beetle is eating are moving through the beetle. Well, then I use that as my signal. So then, as my signal to identify the frass, the feces produced by the beetle. So then I found myself doing this. Take a beetle, well, 96 of them, put them in a 96-well plate with media and fluorescent spheres. Leave them eat there for 4 hours. Take each beetle out, wash the beetle, and put it in a little toilet. Wait for the beetle, check with a microscope and look for the spheres. If the spheres are there, you got your frass. Okay, it worked. And what I'm showing you here is how the frass looks like when you collect it in these little plates. So what we did then is that we compare caffeine concentration in the food and in the poop using different techniques. So that was the first part. You take normal beetles, they go there, you collect the frass. Then I took my beetles, applied a bunch of antibiotics. I see that they are alive, they are well. I take them back to the diet, collect the poop, and measure again. And finally, again, take your beetles, treat them with a bunch of antibiotics, leave them rest for a bit, and then give them back one of the caffeine-degrading microbes. See, prove that when you put that microbe back, or when you increase its density, that now you have caffeine degradation. So that was the experiment. I want to show you the results here from this paper. Our control were our beetles where we did nothing. So if you see here, the levels of caffeine in the frass are 0.34 milligrams per gram of caffeine. When we use GCMS, we also use FTIR. When we treat our beetles with antibiotics, you can see that there is a lot more caffeine in the poop, in the frass. But then when you reinfect them, or reinoculate them, I should say, with Pseudomonas, you again have the degradation of caffeine. So this for us was the evidence to say, well, the microbes are not only active in the beetles. We see them when we amplified cDNA. We look for the signals of who is active. But when you deplete the microbiome of your beetle with antibiotics, and then you give them back a microbe, they regain the capacity to degrade the caffeine. So this was one of the nice examples of an animal, depending on its microbiome, to perform a task like caffeine degradation and become a coffee pest. Okay, I was supposed to use, always fail. What are we doing now? Okay, I had to stop a little bit the project while I was in the Berkeley lab. The Berkeley lab is a laboratory that is funded by the Department of Energy. So as you can imagine, they didn't really care about this type of work, so I stopped it. But now that I'm back at NAU, we are doing the following. We are working again on the project. And what we are doing is that we take beetles from the field. We look for bacteriophages. Do you guys know what a bacteriophage is? It's a virus that will kill bacteria. They are very specific. They don't kill every bacteria. They have their specific host. So now what we are looking for are these viruses within the beetle gut and in soil that will actually kill these bacteria. So when we identify them and we see that they can kill the bacteria that helps the beetle, we are going to try to use them as a treatment in the beetle to get rid of bacteria that are helping the beetle to degrade the caffeine. So that's part of the work that we're doing there. And here you see the faces of the 3 students that now dissect beetles like pros. Like I'm super happy and surprised. I mean, they even get to the point of use microsensors to then measure oxygen, hydrogen, and other properties within the beetle gut. So they are that good. They don't have my shaky hands. So what we have here is actually, yes, A beetle that is a coffee addict and that is completely enabled by its microbiome. And now we are looking for different strategies to actually try to remove those keystone microbes from the beetle gut using very targeted approaches. One of them is the use of bacteriophages to regulate the microbiome of the beetle. We wonder, could this be a new strategy for pest control that could be used out there? Instead of just like spraying chemicals or targeting all microbes, could you identify, all insects, could you identify a microbe within the pest that you want to get rid of and then control that pest? So that's something that we're going to try to continue to work on, and just look at the happy guy there. Okay, so we are good on time here. Well, I'll say, that was the beginning of my work in LBNL. And then I was offered the possibility of going back to my roots, which is working with soil. But now, after working with insects for a while, I thought, well, when you think of soil, everybody thinks of bacteria. Everybody thinks of the fungi. But when you look at soil, if you ever have looked at soil, you will have many other organisms. And here I'm going to start with microarthropods, for example, even nematodes. But when you go a little bit deeper, you can see there that there are protists, there are viruses, so it's a very complex environment. So that changed a little bit the way in which I wanted to study soil. And well, now, and for years now, I'm trying to bring a holistic view of the soil microbiome, in which if I study the soil, I will try and work to study everybody that lives in a soil, or everybody that is microbial, in my view. You can have here the bacteria, you have the archaea, fungi, you can have the viruses that kill bacteria, you can have the protists that eat bacteria, and then to try to get a general view of the soil microbiome. So with this idea of a holistic view, I want you to help me. And I want you to choose your own adventure one more time. What should I tell you about? Should I tell you about microbial food webs and carbon flow? Or should I tell you about the fragility of cross-domain microbial communities? Who wants to hear about microbial food webs and carbon? Nice. Who wants to hear about the fragility of cross-domain microbial communities? Alex, pay attention and tell me which one is it. What do you want to hear about? Yes. The carbon flow. Okay, we'll go with the carbon flow. Okay, give me a moment. So, there. This is a project that is still ongoing, and it's a collaboration between UC Berkeley, the Lawrence Livermore National Laboratory, and Northern Arizona University. As you may know, in soil, nutrients such as carbon and nitrogen are recycled by the activity of microorganisms. Everything will start, for example, with a primary producer like a plant. The plant will fix CO2. The plant that will produce sugars, distribute the sugars and other chemicals into the soil. And then from the soil, microbes will utilize it and create a food web. They will eat each other. They will exchange metabolites with each other. Now, this is what one would expect to happen, right? We have a specific question. Okay, we know that the mechanism could go this way, but how could this mechanism be affected by drought? When you don't have enough water in the soil. In normal conditions, in my nice situation, you will go to the soil environment, you will use a microscope, or you will just make yourself very tiny, and you will see that all communities are interconnected in a way by water. Water will create these channels that will connect communities with each other. You will have microbes that are degrading organic matter, producing metabolites, then the water maybe will help to move the nutrients from one place to the other, fungi as well. But then the water will also work as a channel for the movement of predators from one place to the other, like the protists, the nematodes. They need water to move around. Do you guys know what is a protist? Okay, for those of you that don't know what a protist is, because I love protists, they are eukaryotic microbes. These are microbes that have a nucleus, like us in our cell, but they are so diverse And many of them are predators. They like to eat other organisms like bacteria, like fungi. And when they do that, well, they release a lot of nutrients into the environment. Can we get that out of the way now? Very cool, yes. So everything is connected. And then the idea is that when you have drought, well, those pests get removed. And now communities will be isolated from each other. And maybe the process of carbon cycling will change. That's just a general idea for the presentation that I want you to keep. So what we did is that we went to Hopland, California, and set up a field experiment in which we had treatments in which we reduced water by 50%. We knew how much water was supposed to come into the environment by records, et cetera. So we just applied 50% of that water. And then we had other plots in which we just applied the regular amount of water that the environment is supposed to get. So here you can see that we have our treatments, 50% water, 100% water, regular water, or drought and control. And I need to say here, prior to the first sample, we treated our soils for 5 months of differential water treatment. So there were only 5 months of reduced water treatment. And then there was a question, could a short-term drought have any effect on microbial communities? That was the first question. So again, we went there, collected roots, removed the soil from the roots, extracted DNA, and looked for one gene, just one gene, I will say, that I'm marking there. So we analyzed bacteria, fungi, protists, and also nematodes, little microscopic worms that also eat bacteria or attack plants. And from there, we first wanted to see, are communities affected by our treatment? Maybe this plot looks a little bit strange, but I'll explain to you. So here you have the plot for bacteria, fungi, protists, and nematodes. If things of the same color are overlapping each other, it means that the community composition is very similar. If things are separated from each other, then community composition is very different. What do you guys see there? I saw nothing. So it seems that after 5 months of our treatment, we didn't have a significant effect, at least with this tool that we were using to analyze changes in community composition. So we wonder, are we using the correct tools to define changes in community structure, right? And well, it was sad because we're investing a lot of money in this project, and imagine going to the field and do all those manipulations. That was something, it was only one site. So then, because I love networks, we decided to create networks of drought and normal soils that I want to show you here. Here you have our control. That is the network that includes bacteria, fungi, protists, and even nematodes, metazoa. All that craziness that you see here is how these organisms are co-occurring. Okay, this is not an interaction. Connecting things are not interacting, they are just co-occurring. This is just a snapshot of what we have there. Now, what happened with our drought treatment? That is a network. So while analyzing community composition on its own was showing us nothing in my view, even diversity was showing no effects. When we went to reconstruct the networks, we could see that there was a likely potential effect there in the treatment, only 5 months of drought that we applied. And yeah, they are pretty networks. Here are some numbers for you. You can see the number of nodes, the little dots and links. And I want you just to see the difference between the two networks. If you can see, there is a significant change between the two, or a high change. It seems that the control has a lot more links connecting every node than the drought treatment. From the networks, what we did is that we calculated an index called cohesion. So it just indicates how connected things are. That's all. We can see it there, but a pretty network will say nothing unless you have values to do a statistical test, and well, we can get there. So you can see here that cohesion actually changed significantly between the two treatments. So we saw that something was happening in our network. So it was like, yes, well, at least DOE is not going to kill us for using all this money. So when we applied these treatments, we thought, well, okay, we see it in the networks. What's going on here? What's happening with carbon? So we went back to the field, and actually now, remember that plots that I showed in the field? So we went to the plants, we put a chamber on top of the plant, imagine a jar on top of it, and flushed it with 13 CO2, heavy carbon in CO2. So now the plant could feed that carbon, heavy carbon, get it into the tissue and produce sugars that now are heavy because they have 13C carbon. So our idea was then, how is that carbon moving from the roots all the way to the food web? And we applied what is called QSIP. Simple is my word. You get the roots, you extract the DNA of treated and untreated, you put your DNA in these microcentrifuge tubes, and you can imagine that the DNA of different microbes just arranges within the tube. You apply centrifugation with cesium chloride. And what is going to happen is that after 5 days of centrifuging, there you go, heavy things will migrate lower in the tube. And if something migrates lower in the tube in reference to where it's not labeled, you could say that that microbe was accumulating the heavy carbon. Are we there? So that microbe is eating that 13C that the plant is pumping into the soil. So what we did is that we took that DNA and again looked for one gene. Okay, one gene to characterize all the communities. So you have it there. We analyze bacteria, fungi, and also protists and other organisms. I will go really quick through this, but what I'm showing here is how we detected bacteria in our normal or control and in drought, and the levels of enrichment, how much carbon these microbes had just by using sequencing. We did the same for fungi. You can see there a lot of fungi highly enriched in the control, not many in drought. Protists, the same, the little predators that move through the soil. And finally, the nematodes and other organisms as being highly enriched between our treatments. So now we have an idea of who it's who when we just look at their names. So based on this, I decided to reconstruct a network. Let me explain this really quickly. We have the carbon getting into the plant. You have all the microbes that are enriched. And from here, we could say, okay, the fungi, Penicillium and Bipolaris, could be associated with the plant, so they are likely getting the carbon from the plant. That's why they are labeled. Bacteria that we found in the rhizosphere may be also getting it directly from the rhizosphere. This nematode that I have here, Merlinius, likes to suck the life out of plants. So it's getting the carbon from the plant. But then others are eating each other and getting the carbon by eating other organisms. That is just an empirical network that we created based on what we know, based on what is reported out there from the names of these organisms. But then again, I decided to apply network analysis. If you see, I love it. I love network analysis. And here, we reconstructed now a network with enriched organisms. And what I want you to see here is, in red color, the bacteria, then fungi, protists, and nematodes. And you can see here, for example, that this nematode, Porcelaimellus, is an omnivore, meaning it can eat anything that you put in front of it. So it's potentially co-occurring with bacteria, but also with protists, eating the protist. Other protists that are omnivores as well, they are likely eating each other and eating bacteria. So with this type of technology and sequencing and QSIP, we are trying to reconstruct how the carbon is moving from the plants all the way through the soil. I'm not going to show it here, but we see significant differences in the network of organisms that are sharing the carbon in normal conditions than in drought. So only 5 months of drought have had a very important impact in community composition, by also in the movement of carbon through the food web. Now I want to say, we are working on the paper, but now my student Ivory in the laboratory is helping me to see if any of these interactions that we saw in the DNA and in the field can be reconstructed in the laboratory. One thing is that you see the signals of DNA in the soil, but is this actually happening? So what we are doing in the laboratory is that we go to our soils, we prepare these really crazy enrichments that I want to show you here. If you see there are things that are swimming around. These are the protists. We have also bacteria. We have archaea in these enrichments. You can see the guys there, like moving around. So we prepare these crazy enrichments in the laboratory. We then do metagenomics, analyze the DNA of everything that is in there. And we can identify now that in this enrichment, we have viruses, bacteria, even predatory bacteria. Do you guys know that there is bacteria that kill bacteria even? They will pierce them and eat everything out of them. And of course, in our enrichments, we also have a bunch of proteins. And you can see here how we are being able to reconstruct the genomes of these organisms only from DNA out of our enrichments. So what Ivory is doing now is that she's having experiments in which she removes the protist and add everything else to a plant. Let's see what happens. Now, if you put back the protist, you have a predator there. What is going to happen with carbon and nitrogen cycling? When you add back the viruses, what's going to happen there? That's the questions that we're trying to pursue at the moment. And the idea with this is that in general, we want to understand better how our soils function. This is just another approach to do it. And again, in my view, it's a holistic view. We are trying to be inclusive all over, also in science, which I love. So with this, I'm going to finish my presentation. I want to thank all my collaborators from the past and the present. And here you have the faces of my team at NAU. Oh, we're on time. I was rushing here. We have more time for questions. So thank you very much, and happy to answer any questions. And just to show you this, the other stories were there. Don't think that they weren't there. So I was ready also with the other ones. Okay, so yeah.
Any questions from you?
Hi, can you hear me? Yeah.
Yes.
Great talk. Going back to the first story about the coffee borer, I'm curious about any effects of toxicity when you remove the microbiome from the borer. Like, are you able to see effects on the beetles that the caffeine is now, hurting them in some way? Yeah, exactly. Because you've removed the microbiome that is degrading the caffeine.
Yeah, very nice question. And this is something that we started exploring actually last year. I couldn't do it back in the day, but I will tell you, yes, there is an effect. We see that there is a delay in reproduction. Females are not reproducing as fast. Also, the oxygen or the respiration rates of the beetles are changing. They are actually being what I will call more active in getting the oxygen or respiring faster. So we think that the caffeine is affecting even, the physiology of the insect. We can see that at certain levels of caffeine also, even when they lay eggs and larvae come out, larvae start to dry out. So there is something going on with the beetles, and they are very highly affected by the caffeine content. Now, we are doing this in a laboratory setting. We make our own coffee smoothies for the beetles. And what we do there is that we add pure caffeine into the diet to take it to the levels that we have in the field. So we can see that beetles will survive kind of okay around 5 milligrams per gram of caffeine, which is nothing. I told you that in the field, they are exposing to 20 to 30 milligrams per gram. Then we take them to 10, 20, and then is when things go wild for them. Now, this is with beetles treated with the antibiotics right now, right? So, so still we're exploring a lot about that. Yeah, thank you. Mm-hmm. Yes.
Were you surprised that reinoculation with a single species was enough to get you pretty close down to those caffeine levels that they sort of control?
Definitely.
What did that mean for the applied use of this in coffee?
Yeah, no, absolutely. I mean, we were very surprised. Here's the thing why we selected one. Okay, we selected one because that microbe, which was a Pseudomonas filba, was the one that was degrading caffeine so nicely in liquid media when you only put the pure caffeine in there. So it was just growing like if nothing was happening. So we thought, like, let's use this guy and just put it there and see what happens, And the idea back in the day is that we will test different combinations, but we have to hurry, like at some point. But this makes us or made us think back in the day about the functional redundancy of the microbiome. If you have a microbe that on its own could provide that benefit to the microbe, why is everybody there? Why everybody else there? And will they be able to take that niche if we remove that specific microbe? And what if our phage treatment will not work because everybody else will come and take that place? So these are really cool ecological questions that we still need to think about. It was a big surprise. So yes, yeah, thank you. Yes.
Hey, thank you. Good talk. I think kind of following up to that question, I was going to ask did you think it was like a metabolite where the bacteria was like helping them break down the caffeine, or if it was actually the bacteria itself? And based on what you just said, it seems like the bacteria itself is like processing the caffeine.
Mm-hmm.
And so my question, I guess, following up on that was going to be, and I guess kind of back to other questions about the network analysis, right? You always find this there. And so because it's always there, you presume it's doing something important. But if it's just breaking down the caffeine, presumably there are many other bacteria that could also break down caffeine and could also colonize their gut. So like, why are you finding this everywhere?
No, you know, it's a very good question. Absolutely. I need to say that back when I was doing this work, we focus on the Pseudomonas because it's the one for which the whole pathway of caffeine degradation is already elucidated. For the other ones, we don't know how they are doing it. Now, let me step back a moment. I have the genomes of everybody else, and we are trying to see how they are degrading the caffeine. But you know, in general, I think it's a good question that we still need to pursue. One strategy that we're using now, that we're about to finish, is that we are now analyzing metagenomes, the whole community within the beetle, and metatranscriptomes to see, you know, to try to associate more of activity that is related to different levels of caffeine and maybe try to answer those questions. At this moment, I don't have an answer directly for that, but yeah, I mean, I hope that metagenome and metatranscriptome will give me some of the signals of the activity of other organisms. I can tell you something, that's not the only thing that they are doing there, okay? So they are not only degrading caffeine. We know, for example, that the beetle gut of this tiny insect has oxygenic and anaerobic conditions. So there is a lot of fermentation and other processes happening within the beetle gut. We just don't know because we didn't get to analyze it. So now I hope we will bring some more answers there. Yeah.
Great talk, thank you. So with the idea of applying the bacteriophages to the soil as opposed to pesticide, I know that you said they were pretty specialized, but would you be concerned about those bacteriophages maybe affecting other organisms that are also reliant on those bacteria for different things?
Absolutely, but hear me out. What we know about phage biology is that they are highly specific, even at the level of a strain. You know, so even though you could apply them, they will be affecting a specific group organisms, at least the idea. So it's something that we will have to consider. We are really far from any application. If you see, we have our work in Petri dishes, we have our work in a laboratory setting, and we are at the moment trying to develop a proof of concept type of approach, you know. Getting to the point of spraying phage out in the environment, I mean, we are far from it, but there are a lot of things that still we have to analyze. So it will have to be considered.
Thanks. Such a cool talk, and definitely want to hear the other story we didn't get to hear today. I'm wondering about a couple of things about the drought experiment. And in just sort of trying to wrap my head around what it means for there to be the same community composition but different network structure. Could you say a little bit more about one, if like, I missed if that was DNA or RNA, is that possibly just remnant necromass DNA? And also, if you have any thoughts about sort of spatial co-occurrence, like are things just moving to more basic microsites, or how are they distributed throughout the soil?
Yeah. In the case of about the remnant DNA, you know, or the DNA they just left behind, when we applied the QSIP technology, the idea is that you are getting the signals of organisms that are active at the moment of sampling. You know, so there, at least that's the application of QSIP at the moment, QSIP sequencing. So I will stay away a little bit from that. We believe that is the DNA of active organisms. Now, how are communities segregating? You know, if they are segregating, is it actually happening what we say, you know, that the channels of connection provided by water and soil structure are being disrupted by the environment? That will require a little bit more of a spatial type of resolution analysis. It will be hard to describe, I think, but that will be in part of my hypothesis. You know, things are being isolated from each other due to the removal of those connectivity channels that we may have in the soil. But again, that's an hypothesis that we see in a way translated in our networks derived from the analysis. I'm not saying that this is the final answer to any that we're seeing in the field, but it shows a little bit of signal and hope that our experiment was going well, and that we could move forward with many other things. But yeah, thank you. And we could talk more about this and the other stories if you like. Yes.
Yeah, I was just curious about the bacteriophage treatment
Yes.
idea. Are there examples of that already, or would this be like a first time using that?
There are examples out there. There are different, even in San Francisco, there is a startup that is testing different bacteriophage for the treatment of Clostridium difficile in mice. I know as well that cocktails of phage are being applied in patients where nothing else is working right now, but these are a type of last resort of resource that you could get to use. Yeah, but there are examples. It still is very developmental. I think it's just getting there. I know at the same time that the Department of Energy, for example, is very interested in manipulation of microbial communities in soil. It's not going as fast as one or as they want. So my idea here is, if I can prove that I can do this in a contained system as a beetle that I understand really well, maybe we can move forward into different systems. Our examples are there. They are variations in success, absolutely. But it's an idea. Yeah.
Yeah, great talk. The beetle being a pest, it doesn't play any other terrestrial role as food for another organism. And is there any concerns about, getting rid of them by stopping them from their primary source that, yeah.
We know that ants love to eat these tiny beetles when they get into the soil, because at some point when the harvest is done and there is no more place for them to live, they go into the soil for a little bit. So we know that they are being eaten by other organisms. And yeah, again, you get to the point, what's going to happen when you remove it? I mean, it's a very interesting question in ecology and manipulation in ecology. So it has to be taken into consideration. What other service are they providing, if any? I'll tell you that we don't know. Okay, we don't know. There are a few groups that are actually studying this beetle, mostly for pest control type of approaches, but its ecology in-house providing any benefit into the environment, yeah, it has to be analyzed.
Time for one more question.
Yeah. Absolutely, yeah. On one side, they want us to generate this type of data to improve the resolution of ecological models that we have out there to understand how our environments are going to respond to drought and how carbon is going to be pumped out of the atmosphere or not. The idea as well is then to try to identify what is important in a soil and try to engineer it to keep it going even after, we messed up the environment or after the environment changes. That's one idea. There are other projects related to biofuel production, for example, growing sorghum or switchgrass even, that are going to same type of questions. The idea is to understand how these bioenergy crops adapt to a nutrient-limited environment and how microbes are contributing to it. So there is always an application behind it. Yeah.
All right. Thanks so much.
Thank you. Yes. Ooh. Done.